So today we're going to be talking about DNA replication, and DNA replication is basically the process by which a copy of the DNA in a cell is made before the cell divides. So for example, DNA replication occurs during the S phase of interphase in mitosis. DNA replication is a semi-conservative process. And this basically means that the newly synthesized strand is made using a pre-existing strand as a template. So for example, if we have a DNA sequence, I'm not going to draw the double helix just to make it a little more simple, but if we have a DNA sequence which is basically A, C, G, A, T, we know what the other strand is going to be due to complementary base pairing. So just a reminder, A always pairs with thymine with 3 hydrogen bonds. With 2 hydrogen bonds. And cytosine always pairs with guanine with 3 hydrogen bonds. So because of that principle, we always know that the other strand of DNA is going to be complementary to the original strand. And from that, we can deduce what the sequence is. So using the same principle, if we are given one template, we can essentially synthesize a new strand based on the template, and it would be completely identical to the other strand. And the same thing would happen on this strand. This strand would be identical to this one, and this strand would be identical to this one, due to that whole complementary base pairing principle. So there's many enzymes involved in this process. For the HL syllabus, you need to know all of them. However, for the SL, I believe you only need to know helicase and DNA polymerase 3. So this diagram already has, this diagram already has pretty much everything that you need to know, but I'm going to go into depth into all of the proteins now. Also, another thing about the whole semi-conservative process, this is elaborated further in the Michelson and Stahl section of the DNA unit, which we'll be covering in a different video. So the first protein is helicase, and helicase unwinds and separates the double-stranded DNA by breaking the hydrogen bonds between the base pairs. So again, it breaks the 2 hydrogen bonds between adenine and thymine, or the 3 hydrogen bond between cytosine and guanine. And these hydrogen bonds are basically what are holding the 2 strands of DNA together. So without them, they become separated into 2 strangle strands. And helicase unwinds the DNA at a region called the replication fork. And this essentially produces 2 strands running in anti-parallel directions. So if you refer back to the DNA structure video that I made last week, you would know that DNA naturally is arranged in an anti-parallel structure, meaning that one strand is 5' to 3' and the other strand is 3' to 5'. However, due to this anti-parallel structure, we'll touch on this later with when we talk about DNA polymerase 3, it's a slightly different process for DNA replication based on whether whether we're talking about the leading strand or the lagging strand. Again, don't worry, this will be explained in a moment. So the second enzyme that we need to know is DNA gyrase. And DNA gyrase reduces the torsional strain by the unwinding of DNA by helicase. So it does this by relaxing positive supercoils by negative supercoiling that would otherwise form during the unwinding of DNA by helicase. So it basically twists the DNA in the opposite direction to prevent it from forming back into that double helix structure and like twirling or curling essentially. Then we have the SSB proteins, which are the single-stranded binding proteins. And SSB proteins bind to the DNA strands after they've been separated to prevent the strands from reannealing. And again, reannealing is just the process by which the 2 strands of DNA return back into that double helix structure. These proteins also prevent the single-stranded DNA from being digested by nucleases or enzymes which digest nucleic acids. SSB proteins will become dislodged from the strand when a new complementary strand is synthesized by DNA polymerase 3, which again I will talk about later. Fourthly, we have DNA primase, and DNA primase basically creates short binding sites for the DNA polymerase 3. It creates RNA primers on the template strand, as you can see here. And this RNA primer is essentially an initiation point for the DNA polymerase to start its nucleotide chain. So again, DNA polymerase can't begin its own nucleotide chain, but it can only extend one. So DNA primase makes those initiation points. Then we have DNA polymerase 3. So free nucleotides align opposite their complementary base partners, which is again A to T and G to C. DNA polymerase 3 attaches to the 3' end of the RNA primer and covalently joins the free nucleotides together in a 5' to 3' direction. So in this strand here, you can see that it's working in this direction, whereas in the top strand, it's working in this direction. And this actually results in what we call a leading strand and a lagging strand. So this is denoted as the leading strand. And that is because DNA polymerase 3 is moving towards the replication fork and can synthesize continually. Because if you think about it, as helicase, which is the one that separates the DNA, continues to move forward, this line will just keep extending. This DNA will just continue to get separated, and DNA polymerase 3 can continue to work in a straight line. However, on the lagging strand, which is this bottom strand, DNA polymerase 3 is moving in the opposite direction, away from the replication fork. So basically, what it has to do is jump back constantly and replicate the DNA in little segments that we call Okazaki fragments. So again, this is due to the anti-parallel nature of the DNA, because one of them is running in a 5' to 3' direction, whereas the other one is running in a 3' to 5' direction. However, DNA polymerase 3 can only work in a 5' to 3' direction. So that results in two separate methods of DNA replication based on which strand is being replicated. Next, we have DNA polymerase 1. As the lagging strand is synthesized in Okazaki fragments, it has many of these RNA primers just on the strand. For the leading strand, we only need one RNA primer at the very beginning. After that, it's just DNA polymerase 3 that continues to extend it. However, on the lagging strand, we have multiple, and because of that, DNA polymerase 1 is needed to replace these RNA primers with DNA nucleotides. Then finally, we have DNA ligase, and DNA ligase joins these Okazaki fragments together on the lagging strand to form a continuous strand, and it's now exactly the same as the leading strand. It does this by covalently joining the sugar phosphate backbones together with a phosphodiester bond. Just another thing about DNA polymerase 3, with the free nucleotides, they are found naturally with 3 phosphate groups. So DNA polymerase 3 cleaves 2 of these phosphate groups and uses the energy released from this process in order to join the nucleotides together and form phosphodiester bonds between them. So yeah, that's the process of DNA replication. There's lots of enzymes involved. However, the process should be quite logical if you think about the reasons why every step is happening. Like, why do we need to separate the DNA? Oh, so it's that we can expose the nucleotides so that they can be replicated. You know, why do we need DNA gyrase to prevent it from reannealing so that it's to prevent it from recoiling and reducing the torsional strain so that it doesn't form that double helix structure again. So yeah, hopefully this video was helpful. Thank you.